same. It's characterized by the blood glucose level goes up, then the insulin kicks in and brings it back down. So how well can one do if one practices what I call the context-relevant approach to managing the madness? This is actually me wearing a continuous glucose monitor for about a week. And you can see that almost the entire time my blood glucose level is between 80 and 140. But the amount of work that went into this was phenomenal. I struggle with trying to teach my patients everything that they need to know to try to achieve this kind of control. And I don't have a good answer. It's very difficult for me to impart all of this knowledge. You can see that in a 1 week period of time, I actually was above 200 for about 4 to 5 hours. But normal in well-controlled diabetes is about 6 hours per day. So what about free fatty acids? I could talk about the structure of free fatty acids. They're basically a form of fat. And when 3 free fatty acids are combined with this glycerol molecule, it forms triglyceride, which insulin then promotes storage into fat cells. I could talk about the molecular mechanisms whereby free fatty acids actually reduce our responsiveness to insulin, or how free fatty acids cause the liver to dump out even more glucose. But to simplify things, let me just say that free fatty acids are not free at all. They come at a great cost. They cause insulin resistance. They cause blood glucose levels to go up, and they cause us to have to take more insulin. And wouldn't you know it, that free fatty acid levels are elevated in type 1 diabetes. And they're all over the place like glucose is. So here was a study that was looking at free fatty acid levels after a meal in people with type 1 diabetes and in normal people. And in normal people, you can see that free fatty acid levels in response to insulin go down a little bit and then return to normal. But they're perturbed in type 1 diabetes. They don't suppress normally in response to insulin, and after some period of time, their levels actually go very high. And so you have a system where these free fatty acids are changing how sensitive we are to insulin that is not regulated in the first place, and it becomes very easy then to understand why blood glucose levels in type 1 diabetes are such a chaotic mess. So what about leptin? You've heard so much about it, and leptin is actually a natural hormone made by fat cells. These are 2 totally insulin deficient mice, and this was work done in the lab of Roger Unger. So it's a mouse model of type 1 diabetes. These mice totally lack insulin. And you'll see 2 mice. The one on the left is being treated with a saline solution that really doesn't do anything. It's just a control. The mouse on the right has no insulin, but is being treated with a level of insulin that's just a little bit higher than what the body would make normally. The mouse on the left is a sick mouse. Its blood glucose level is in excess of 600. It is ketoacidotic, and it will die. The mouse on the right is a happy mouse. It's healthy, it's growing normally, and its blood glucose level is normal without insulin. And so not since the discovery of insulin in 1921 have we believed that there was a hormone other than insulin with this capability. What about blood glucose control in rodent models treated with leptin? And again, this was work done in the lab of Dr. Unger. And first what you will see is my attempt to just keep a diabetic mouse alive by treating it with a long-acting insulin analog that I thought would hopefully normalize the blood glucose level to some extent. But you will see that it's all over the place all of the time. And so it's very much like type 1 diabetes. But in a set of experiments done by Mei-Ying Wang in Dr. Unger's lab, when she gave leptin combined with a dose of insulin that was reduced by 90%, this was the blood glucose profile achieved. And this is exactly what we're going for in the human trials, now testing leptin in type 1 diabetes. So how does it do this? How does leptin stabilize blood glucose control? Well, I've talked about the 4 things that cause variability in glucose. I talked about food, I talked about glucagon, I talked about our dependence upon injected insulin, and free fatty acids. And so the first thing that we know is that leptin, the hormone, suppresses appetite. It actually signals through the brain to make you feel full. And so, in fact, it is being developed as an anti-obesity agent by Amylin Pharmaceuticals in conjunction with Takeda. Leptin also normalizes the glucagon elevation that typifies diabetes. So here is one example of the extent to which glucagon levels can be elevated in a type 1 diabetic mouse model relative to normal, and the extent to which leptin levels can be glucagon levels can be normalized by leptin. And this is a very complex set of slides that is helping us to begin to understand how it is that leptin and insulin are similar. This is a metabolomic profile of livers from mice that are either normal or diabetic or treated with insulin or leptin. We send these to a guy at Duke, Chris Newgard, who does this detailed analysis that really helps us to begin to understand what exactly it is that leptin is doing. And I could go into great detail here, and you would probably think that we're all very smart, but you probably wouldn't understand a lot. So let me simply say that what we're beginning to understand is that insulin and leptin are really quite similar in these rodent models in terms of how they impact metabolism. More fancy science here, but the bottom line is that what we know is that insulin and leptin differ in one very important regard. Whereas insulin promotes the formation of fat and cholesterol, leptin actually suppresses their production. And so if we consider uncontrolled type 1 diabetes, we know that the system is flooded with glucose and free fatty acids. And if we treat with insulin, glucose moves into the muscle cells where it's utilized as a fuel source, and free fatty acids are moved into the fat cell compartment, which is a safe storage compartment. But there they remain to flood the system again and wreak metabolic havoc when insulin levels fall low. On the other hand, if we consider type 1 diabetes treated with leptin, what we know about leptin is that it actually oxidizes fatty acids. It literally leads to their burning such that they leave the body as CO2 and water and leaves the fat cell compartment depleted. And so, to summarize, what we know is that insulin and leptin have many similarities in terms of how they impact the metabolism of glucose and proteins. But whereas insulin promotes lipid and cholesterol production, leptin is actually lipid depleting. And when we think about the things that cause the glucose roller coaster, we've talked about food, glucagon, insulin, and free fatty acids. And what we know about leptin in these rodent models is that it reduces food intake, glucagon levels, a dependence upon non-regulated injected insulin, and free fatty acids. And so what we are hoping for in the short term as we begin testing leptin in humans with type 1 diabetes is that it will reduce the daily burden of managing the disease and get us off the glucose roller coaster. And in the long term, it will help to reduce complications such as heart disease. And so we have begun the first clinical trial. We have 3 patients who are already being treated with leptin. And unfortunately, I can't talk about the results of the clinical trial as of yet, but I can tell you that we need more subjects. We're looking for young adults who are thin and have a hemoglobin A1C of somewhere between 7.5 and 9. It's a 7-month study altogether, and 5 of those months include being treated with leptin.
Define young adult. Define young adult. What age?
18 to 50.
Makes us feel good. I'm young!
So this slide is meant to remind me that, you know, anytime you hear me talking about my research, whether it was stem cell research or pancreas regeneration research or leptin research, you'd probably think that I'm going to be able to cure you of your diabetes tomorrow. Nothing could be further from the reality. It takes much time and much effort to bring these things to fruition. I remain committed, and I hope that you will too. And in the meantime, all I can say is keep running. I'd like to say thank you to especially the members of the local chapter of the JDRF who have been so supportive of our endeavors, who went to great lengths to organize a press conference with the mayor of Dallas to help us recruit people for participation in the study, and just for your all-around general support. It takes a huge team of people for something like this to happen, and I feel very fortunate to be right in the middle of so much brain trust. Chris Newgard from Duke has really done some phenomenal work that has helped us to begin to understand the science of leptin in these rodent models. David Maggs, Elaine Chiquette from Amylin Pharmaceuticals, we've worked with very closely. You know, they're approaching this problem from a perspective that we would never even imagine in an academic institution. And it gives me great hope that if we are successful, that our ability to bring this to the marketplace will be enhanced. Jeff Friedman is the guy who discovered leptin and who has been so supportive of this project, as has Michael Brown. Mike Brown is one of the Nobel laureates at UT Southwestern who has believed in this project from day one and has used all of his power and reputation to try to move it forward. Abhimanyu Garg is phenomenal. Abhimanyu Garg has studied leptin in human diseases for over 10 years at UT Southwestern. And so he has in place this entire infrastructure that he is allowing us to use to test leptin's potential in type 1 diabetes. Sanjoy Dutta from the JDRF is a person that we've worked closely with. And everything we owe actually to Roger Unger. Roger Unger is the creative genius behind all of this. And so if it is successful, we will owe it all to him. With that, I'd like to just say, hope to see you all at Dream Gala. I will be dancing hard. And thank you.